Well, good afternoon, and thank you very much for the invitation. Our subject today is pain, a sensory experience which everyone in this room has had and probably has quite often. But it's a subject that is particularly interesting for many different reasons. For example, pain has a way of capturing your attention. If somebody steps on your toe, or you're playing tennis and run and land on a thumbtack, you stop everything and deal with that. It captures your mind. It captures whatever is going on in your head. I think it's probably the closest sensory phenomenon we have to conscious experience and to the circuitry that is involved in conscious experience. And it's an experimental entree into this subject, but that's not my topic today. Another of the interesting features of pain is that it's very common, probably the most common and the most problematic and the most expensive of any of the biomedical problems having to do with the neurosciences. So, for example, my guess, I don't know, but my guess is, just according to the numbers, there's nobody here in the room with Parkinsonism, and probably nobody here with ALS. However, I venture a guess that more than just a few have at some occasion or another had back pain, or maybe arthritis, or maybe particularly unpleasant menstrual pains. In fact, according to the statistics, probably 50 people for having one sort or another of chronic pain, which may include, as I said, arthritis or migraines or many others. And despite the very common nature of these problems and the very high costs involved, we're living in a previous century in terms of our ability to treat and the drugs we have available to treat. So the most common drugs, the ones that a doctor will very likely give you, is extract of willow bark, that is Acamol or aspirin-like things, NSAID-type drugs, or things that are even older, the juice of poppies, opiates of one sort or another. And only if you've got a very special kind of pain, you might be given an anti-epileptic drug, something that was developed for epilepsy but is used to treat pain, or perhaps an antidepressant. And we almost don't have anything that was developed for the purpose of pain, despite the very great importance of the subject. But that's also not my subject today, but rather variability. And in this, pain really stands out from other sensory systems. So let's start with this picture. We've all been there. And for most of us, I think probably for all of us today, the sky is a light blue, and the dome of the rock is covered with a golden dome, and we'll all more or less agree to that. However, and the same is probably true of taste. So the strawberry is more or less the same from day to day. The same is true of Mozart's music. But when it comes to pain, there's tremendous individuality in the report and the experience of pain due to noxious stimuli or injuries or disease. Even when the injury is identical, I'm going to be focusing particularly on neuropathic pain, which means pain related to nerve injuries. Here's an example, and a classic case of neuropathic pain. Here we're looking at a very large survey done on amputees. So in the sense that it's all the nerves in the limb have been cut across, the nature of the injury is quite similar from individual to individual. And yet, some third of them have quite severe and quite almost continuous phantom limb pain, something over 20 days out of a month. Whereas another third only have pain 2 or 3 or 4 days during the month and not more than that. And some, a fair fraction, hardly have any phantom limb pain at all. So the injury is very much the same, but the pain response is very different. We all know the funny stories about Norwegians who can stand anything, Stoics versus the Italians who scream and shout when anything hurts. And there is some truth to these stories, and it's our common experience with the variability among us and among societies, which has brought over the years the conclusion that individual and group variability is typically attributed to psychosocial influences, to upbringing, to culture, in short, to environmental causes. What I'm going to show you today is that there is an accumulating body of evidence pointing to the fact that there are genetic differences which can account for a certain amount of this variability, and as we'll see, perhaps quite a lot of the variability. Before we get into the toughest, why is it even important to ask the question? And I think there are several reasons. The first, and one that I think is the fact that people who have more pain than others think they ought to have with a particular injury or a particular medical condition are often stigmatized as thinkers, trying to get your husband to do the work, and so forth. And it's true, some individuals, that may be the problem, but I think in many, it's likely to be a problem that aren't your fault. And if society knows, or if an individual knows, that there is a genetics to pain susceptibility, and we're not responsible for our genes, of course, this can help, to a certain extent at least, to reduce or to moderate the unfair stigma. Genetic testing in principle can help us with diagnoses and prognoses of particular conditions and treatments. There's a real hope that one might be able to have an individualized medicine, that is a blood test which will tell you which drug might be more effective for you, rather than just running through the whole series until you hopefully find one that works. From my point of view, of course, the most important potential outcome of this line of work, this question that we'll be asking, is the possibility that we may be able to open new chapters in understanding the physiology of pain, and particularly chronic pain. Now, it's expected that physical traits will be inherited by genetic signals. But how about predispositions and sensory and emotional experiences, which is what pain really is? So I love to look at cartoons. So the doctor is saying, of course I'm sure it's hereditary. My father treated your father the very same thing. So for the medical condition, okay, so that's, there are hereditary predispositions. But is there really a hereditary predisposition to going to medical school? Why do we think that pain predisposition is at least partly connected to genetic factors? Well, firstly, it runs in families and societies. That, of course, is a pretty weak argument, because lots of things run in families and societies that have nothing to do with the genes. But there are twins between monozygotic twins is as expected, or is much higher than between dizygotic twins or simple siblings. And that more points, or at least historically pointed in the direction of genetics. Animal models, which we'll be talking about today, give us convincing evidence that there are important genetic factors. Mutations in humans, which can have very strong effect on pain predisposition, and more recently genetic which really convince us that there are genetic factors and bring us closer to finding out what the genes might be. Of what do I mean by pain? Genes are polymorphisms, and that means variations in the sequence. The expression of the gene's protein product or its function in a way that affects the response.
It's not the gene that is responsible for pain.
The genes account for ability. We all have the same genes, but the variance of the genes can make us more or less susceptible. That's what I mean by pain gene. Now, one could say all of our genes actually go into building our body in certain ways. You need a brain to feel pain, therefore every gene is a pain gene. So that's not what I mean. Or genes are used as tracers or as markers in scientific research. I'm not talking about genetic polymorphisms for variability, for individual differences and population differences. Okay? So this is actually where the question, at least the scientific or the animal experimentation on genetics start here. And this was done in middle of 1987. Peter, we were in the scene then, long time ago. And even what we have here is a model of deafferentation pain, very much like phantom limb pain in animals, where here's a measure of the pain behavior expressed, and this is a measure of one limb. And what you can see is that some individual animals, and each symbol is an individual animal, some of the animals achieve very high scores, that is, they're pain prone, and other individuals are not pain prone, they hardly develop any pain behavior at all, which of course is very much like we know from the clinical experience. So what we did back then, more than 20 years ago, is to take individual pain-prone animals and individual pain-protected animals and mate them together. So here are the high animals, the animals that are pain-prone, and when we take a high male and a high female and breed them, the offspring, when they grow up and we injure their nerves and test them, that is, phenotype them, they tend towards the middle. But if we keep doing that for a couple of generations, if each generation chose the highs among the highs to breed and the lows among the lows to breed, you can see that after 5 or 6 generations, there's a very strong separation of these two groups, and actually the variability, the variance within each group goes almost to zero. This is a very strong piece of evidence that in fact, there is a heritability of the trait for this particular pain phenotype.
So you continue breeding?
Yeah, we actually have them now. We're up to something like 40 different generations.
So you continue the same pain-prone and pain-protected, you continue breeding and they separate?
Yeah, it doesn't take too long. It's just a couple of generations, and they separate very, very strongly.
No, you breed them here, you mix this.
Yeah, the lows are bred with the lows, male and female lows, and male and female highs.
So
Yeah,
you
if
don't
that wasn't clear, then
mix,
we'll get to that in a moment as well.
No,
We can do
because
that as well. If I misspoke, I apologize.
What is
These are high males and high females that are bred together. The offspring of that breeding puts them somewhere in the middle, on average.
why should
But
they?
if we then take those offspring, check them, take the highest among them, and breed them again together, then the next generation, so these are the grandparents, these are the grandchildren, and so forth. So they separate quite quickly. After only a couple of generations, there's a strong separation.
So you introduce an artificial selection.
Selection pressure, experimental selection pressure, yes.
And the rats were not inbred to begin with.
The rats were outbred to begin with. So there was room for they're inbred now. Once we have the separation, we've inbred them. But at the beginning, there were regular subra animals. So there's lots of room for genetic variability among them. That's what I showed at the beginning. So of the regular subra animals, some are high and some are low, and some are in the middle. Okay, let's go on. So we haven't actually pursued this very far, although we do know that the gene or a gene that has a powerful effect on this difference in phenotype sits on rat chromosome 2. But we don't know exactly where the gene is. Now, together with our Larvasi of the genetics department, we went ahead with this program with the aim of seeing if we could find what the actual gene was. And I'm going to tell you about that later on. But let me just introduce the method, which was to take, in this case, 12 different inbred strains of mice and phenotype them on the same pain phenotype that I mentioned before. And what you can see is that some of the strains, like these 3 strains here, have very, very low likelihood of developing pain behavior, whereas others of the strains show very high pain behavior. Now, it's possible to calculate from this a parameter called heritability, which is as it sounds. So the within strain variability. So these animals, for example, this strain here, C3H, all of those animals are clones. They're all identical to one another. So there's no genetic variability there. So to the extent that there is variability in the phenotype, that must be environmental variability. And the same is true, of course, of the ones that are low. So if we just divide the genetic variability by the total, we get a measure of how much of the variability among these 12 strains comes from genetic effects. And the calculation works out to be something like 60% of the variance in this phenotype, in this 12 strains, is determined by genes rather than environment. Now, we then took a very low strain and a very high strain and worked with them. So these are mice that are inbred from the beginning. We're not talking about the selection pressure rats. We're talking about mice that begin as inbred mice. Now what we can do is cross these animals, and we did that with the rats as well, for what it's worth. So here I'm plotting the phenotype, low pain, high pain. These are the low strain animals, 58 inbred mice. And you can see that almost all of these animals are very low. Actually, all the animals are very low. And here, almost all the high animals are high. That's what these columns mean. But when we now breed them with one another and get F1 generation, you can see almost all of them are low. So the gene we're talking about gives a low phenotype and is dominant over the high, if there is a single gene involved. Now we can take the F1s and backcross them, breed them back to the parental lows. And this is the result. All of them are low. And if we breed them back to the parental highs, what we find is about half of them are low and half of them are high. That is a bimodal distribution. And that points towards a Mendelian gene, that is a gene of very high effect, if you just remember your high school genetics. We now know what the gene is, and I'll tell you later on how we know it and how we got that information. If we look at a whole variety of different pain phenotypes, response to heat stimuli, response to pinpricks and things of that sort, that's what all these mean, and use the same calculation of variability that I showed you a moment ago, on average, something like 50% across these different pain phenotypes can be attributed to genetic variants in these various phenotypes. And that's a lot. Now, pain is defined by Mayador, by...
How
Sure.
do you discriminate whether it's the pain that has a higher or lower reading versus the behavior that's accepted?
I'm just talking about the readout, which is a pain behavior. I have no way of asking the rats independently. They show this behavior, which I interpret for many reasons, which I don't want to go into. For many reasons, I think they're feeling the same phantom pain that a human feels with the same nerve injury. But to prove that would take actually another lecture, because there are a lot of different things that go into it. Response to drugs and so forth. Maybe the most interesting thing will be at the very end, where I'll show you that the mouse pain gene here is also relevant to pain in humans. But that'll be at the end. So pain is an experience. It's a conscious experience, a sensory experience. But genes code for proteins, not for experiences, not for sensory experiences. So how do we understand how a variant in a gene sequence, which will change either the expression or the behavior of the protein product, how can we explain that affects how you respond to a noxious stimulus? There are two general approaches, or two general possibilities. The one possibility is that the protein itself is integrally involved in the pain processing or pain generation, generating the signal. And here's an example. So in this particular sodium channel, NAV 1.7, it turns out that a particular mutation in families, rare families, but in families, human families, causes a very painful disease called familial erythromelalgia. It turns out that a different mutation in exactly the same gene that expresses a particular one of the 9 sodium channels, but a different mutation causes other rare families to be totally, or at least individuals within those families, to be totally insensitive to pain. So there was one kid it began actually with the discovery of this family, this kid in where was it? In Pakistan, who used to entertain friends and collect money on the street by stabbing himself and causing all sorts of injuries without pain. Before they were actually able to do genetics on him, he jumped off a building to show that he wouldn't hurt, and it didn't hurt, but it killed him. Now, the other approach, or the other possibility, is that by very indirect means, that a gene polymorphism can affect how one develops and thereby affect predisposition. So the effects are usually much smaller, but much more common. So in our audience here, everybody is likely to have a different sort of predisposition to one pain or another, but we're never going to see this condition. It's an extremely rare condition. Okay, so how would this look? It's not just pain. There is a gene that associates with generosity. I think we've probably heard about that in this room. A gene for risk-taking, for more sex partners. I wonder who in the room has that one. For altruism, for good parenting, attachment, smoking, maternal emotions, extroversion, perfectionism, gambling. One of our own, Dick Epstein, is one of the leaders in this literature. Now, hold on. Is somebody pulling a fast one on this? Is there really a gene for gambling? Let me show you how it probably works. So I'm going to talk about a gene, protein, that has an effect on a muscle protein and has that effect on people who have that protein. That's no problem, because genes and proteins and muscles, That fits together. That's all physical. There's no special problem. However, if you happen to look like that, you'll have more opportunities for that. And if you have more opportunities for that, you are likely to have more self-confidence, more outgoing, and you may involve yourself in sports like that. And if you involve yourself in sports like that, or like that, then you're likely to get hurt. So our muscle protein is a risk-taking protein. Okay? Do you see where I'm going with this? So if you're fightful, you're likely to have that. So the same muscle protein, the same polymorphism, which affects muscle development, is also a pain susceptibility gene. Okay? So not every pain susceptibility gene is going to be interesting in terms of the direct mechanism. In fact, the link may be very, very indirect. But we hope that some will be, and will open vistas for us in understanding them. Now, humans are very, very much alike genetically. The estimates are that 99% of our sequences are identical. That's much more identity than there is the difference for what's worth. So the difference here, I think it's the entire sequence, sir. It even is a tremendous amount of similarity. That means only 0.1% difference among any two random people on average. And 0.1% difference means 1 SNP per 1,000 base pairs. Now, nobody in this room knows what a SNP is, so just have a very, very coarse genetics to see what a SNP is. So here's Tina's DNA. There's a sequence. It goes on for 3 billion base pairs. And there's a sequence, and a certain segment of that is transcribed into RNA and translated into a protein. So that I bet we all know. Now, here's Jan's DNA, and it's identical, 99.9% identical. But you can probably see that there is one difference here in the sequence of base pairs. There it is right there. So a single nucleotide is different. So that's a single nucleotide polymorphism, or its friends call it a SNP. So if 0.1% of the 3 billion base pairs that make up the human genome are different, are polymorphic among individuals in human populations, that means we've got something like 3 million SNPs that can vary and can change proteins and can change behavior ultimately. If it's a rare polymorphism, we call it a mutation. If it's more common than 1%, it's just called a polymorphism. Now, there are classic methods, tried and true methods for finding the polymorphism that's responsible for a particular problem. So the classic method, which has been around for a long time, or only recently has become so efficient that relatively easily, one can find the responsible gene, is by studying linkage in families where certain individuals have a disease or a condition, and then one studies using well-known methods and identifies what the polymorphism is that's responsible for this. A second method that has become very popular in the last few years is what's called association, where we have two populations of unrelated people, where one population has the condition, TMJ in this case, and the other population doesn't have the condition. Now the procedure of finding what is the gene is fairly straightforward in principle. One begins with a candidate, say, this particular SNP here, SNP number 2,245. There's a SNP out of the 3 million. And I just ask, the SNP has, this locus has two variations, an A and a B allele. And one simply asks, is the breakdown in these individuals for A and B, is it any different from the breakdown of this population? And one can simply do a chi-square analysis. Is this ratio different from this ratio? And it's not. However, if I take another SNP, this one here, 4,583, and I see that in the ones that have the pain problem, the large majority have the B allele. And the ones that don't have pain, the large majority have the A allele. And again, I do a calculation. It's very simple, and the statistics show that this is significant. Now I can say that this one, this particular SNP associates with the pain. Now, as you know, if you repeat such tests over and over, you have to do a correction for multiple testing on your statistics, but the principle remains the same. So that's basically how it's done. Now, there are important limitations on linkage analysis. Firstly, it ought to be a gene. You're not going to see it if it's on a gene of very major effect that some individuals in the family have. And when you're talking about neuropathic pain, it's not a spontaneously occurring thing. You'd have to have an amputation. And we're not likely to find large numbers of families where every member of the family has had a particular kind of amputation. So this method really is restricted to very, very powerful, very important method, but very limited. And this is a method which has the general power, and we hope will move us forward. Now, sorry?
Just to understand something. So you're not saying that the polymorphism in this SNP is important for the actual pain or no pain, but it's a marker for the position where the gene is.
No. It's a real change, and if it's in a gene, or even if it's not in a gene, it may regulate the gene. It's something that will have a real effect on that person's body. Now, whether it's a very direct effect, like that sodium channel I mentioned, or whether it's an indirect effect, that's something that has to be studied afterwards.
But he has a point. SNPs are not necessarily occurring in effective changes.
True. But they'll be nearby. True, that's right. Okay, now there are 3 million genes, 3 million SNPs, and finding the one that counts is not so easy. But the fact is that that's not the problem, because we have a technology for checking large numbers of SNPs simultaneously, and it's not that hard to do. So, do we continue with the candidates, as picking candidate SNPs, as I showed you before, and testing one at a time, the ones that, because of our understanding of the physiology, are likely to be useful? Or do we use another method where we can just check all 3 million at the same time? And then you can choose among the 3 million. The trouble is you're checking a lot of different polymorphisms, and you have to have a lot of samples to do the test on. So looking for candidates, we're looking under the light, and this depends on our understanding of pain physiology, and we're not likely in this way to come up with new ideas or new avenues we never thought of before. But the whole genome approach, what's called GWAS, or genome-wide association studies, these allow us to scan all of the genes more or less simultaneously and without a predisposed bias. And that's sort of the true discovery. The difficulty is that they're quite expensive to do, because you have to really have thousands of individuals in the two cohorts, and the costs are very high. In fact, in the pain field, we only have one such study so far.
A couple of hundreds of enough.
No, it's not. It's not. You have to have very large numbers, and if you want calculations, that's Ariel. He's an expert on just exactly that question. So, how have we been doing so far using this method, or using the two methods available? So far, as I mentioned, we only have one GWAS study, and that was published just a few months ago. I'll mention that in a moment. The problem is basically just the cost. The technology is available. There are many GWAS studies in other fields, but so far only one that's asked questions about pain. But Ariel and I really developed an approach which can shortcut that by a great deal by applying a GWAS, a whole genome strategy, to animals, finding candidates, and then asking whether this candidate is available in humans. And this turned out to work very well, and I think we're the first one who have done this, at least in the pain arena. So many important and interesting genes have been found using family linkage studies, but again, these families and these individuals are very rare, and it's not at all clear that the discoveries in these families will translate into more common conditions that are relevant to anyone except them. Excuse me. A few successful association studies have been published already. There's always a problem with replications, but things are moving along. And let me just review some of the things that have been done so far. Congenital insensitivity to pain with anhidrosis, actually quite a long time ago, has been localized to the NGF system, nerve growth factor system, either the growth factor itself or the receptor. And we know why, because if you don't have the growth factor, the nociceptive neurons die shortly after birth, and then you're left without any nociceptors in your body. Familial hemiplegic migraine, a rare form of migraine, 4 different types. The first one depends on, or is caused by, particular mutations in one of the calcium channels. In fact, at this stage, since one studies many families, there are more than 40 to 50 distinct mutations that can cause this condition, or slight variations in condition. And then there are other genes where there are also many different mutations seen in different families. The whole family of Charcot-Marie-Tooth neuropathies, 28 different genes have been discovered up till now which can cause this sort of neuropathies. Some of them, only a few of them, are painful, and some of the genes are interesting ones, like myelin-associated proteins. The primary erythromelalgia, which I mentioned earlier, where a particular mutation causes this very, very intense pain, but a different mutation causes insensitivity to pain. And as I mentioned, that's mutations in the NaV1.7 subtype of sodium channel. That's mostly what we have from familial studies. Association studies. COMT, catechol O-methyltransferase, has been a very popular one. Very difficult to replicate the original results. It often happens that great fanfare with the first report, and then people have a hard time replicating it. It's also been claimed to associate with anxiety and autism and schizophrenia. There, too, there's trouble replicating. So I'm not sure exactly where that leads us.
Marshall, when you say trouble replicating, does it imply the experiment were not really properly done?
Well, or that the N wasn't large enough.
Okay.
What you said is right. The experiment wasn't done well enough. Presumably, if the experiment is done well enough, the discovery will be made, which can be replicated because it's part of nature. MC1R is an interesting one. It's a melanocortin receptor. That's a pigment cell receptor. And it turns out to affect pain response only in red-headed women. It's an interesting one. TRPV1 villanite receptor, which is one of the heat and acid receptors. Opiate receptors, that's polymorphisms with opiate receptors, not too unexpected, can affect pain susceptibility. And it turns out that same NaV1.7 can also be picked up in more common pain conditions in association of these populations. Just continuing down the list, and this is almost a complete list. GCH1, which I'll mention again in just a moment, another one of the TRP receptors. CACGN2, which is a second or a different calcium channel, and all of that in a moment because that's our work. A potassium channel subunit. And this is the result, actually, we're not sure. This is the result of a GWAS study where they're not sure whether it's this gene or this gene, polymorphism is nearby, and they haven't been able to work out yet which of the genes is the key one. Now, what I wanted to point out is these two. The reason that they're special is that both of them were discovered using animal models that were then taken over into human studies. And secondly, that they're both novel. That is, pain physiology at the moment of publication, both of these are quite new. The physiology of pain, or sensory systems in general, would never have predicted these two genes with systems these two genes associated with. And what I'm going to focus on is this one, because that's our own work. And it came from a study or an approach which we call a hybrid mouse-human design, which has really three steps, if you will. First, identify mouse strains, contrast in a pain phenotype of choice. And I already showed you how we did that, and we chose those two strains, C3H and CT8, one high and one low, and I showed you the crossing and so forth. So we already had that worked out. And then use a whole genome method approach to try to find out what polymorphisms are there in these two populations which associate with pain phenotype. And then take that or those genes as candidates to go and test in a human association study. So since we're not testing 3 million genes, but only one or perhaps few, then we can get away with much lower ends and much more acceptable costs. So that's the general approach. Let's see how it works. It has the advantage, of course, this approach that I'm presenting has the advantage that the whole genome or genome-wide screen, so it's unbiased in that sense, and we can have real discovery here. But the costs are something that can be supported in principle, in practice, here at EB University. Another important advantage is here we're in control of the lesion. So we can cause a particular uniform lesion across all the animals we test, where in human association studies you would have to take large cohorts of people, and there's a lot of variability among people. So there are a number of advantages if it works. Let's see how it works in practice. So it all began really with a study of a colleague of mine who used to be here and is now in Toronto, Zev Seltzer, who used an approach called recombinant inbred strains, and I won't go into exactly how that works, but it's a fairly simple and interesting method. And he found that in two strains that were also high and low in the same phenotype, not our strains, two different strains, he found using this approach that here, somewhere in the middle of chromosome 15, there is a marker, what's called QTL, a trait locus, which is associated with, to a different pain phenotype. So it's indicating that somewhere in here, probably a polymorphism, a gene which controls or which has an important effect on this pain phenotype. So our outline began by trying to verify this in using a totally different method, a method of method called microsatellite markers, in a different set of animals, in R2 strains, not strains that substitutes. And in fact, in the entire genome, only one site, one quantitative trait locus, which was linked to this behavior and was in this very same location. So that's our sign that we're on the right track. There's a problem. The QTL marks out a fairly large chunk of DNA, as you saw there, something like a third or a quarter of an entire chromosome. And within that region, about 25 centimorgans, which translates to something like 50 million base pairs, there lie something like 800 different genes. So we're ahead, we've gone forward, but still there's too many. We haven't got a gene, we've got 800 genes, and we have to find some way to narrow down that number. And we used a number of strategies, beginning with fine mapping. Fine mapping was two strategies that Ariel himself actually developed, one called recombinant progeny testing and the second called recombinant inbred segregation test, or RIST. I'll say something about this one. We'll just jump over the other one, but it's published and you can read about it. So that's how the RPT approach works. So it's fairly straightforward. We begin with our two mouse strains, one low in pain and one high in pain. And we make crosses. So these animals are all identical. They've got one chromosome from one parent and one chromosome from the other parent. We now back cross them to the high parents, and we get variability. So if we can imagine this is our chromosome 15, then these are variants of crossovers. Each individual will have a different variant. Now the trick is to find individual animals, like this animal and this animal, where there's a break between the blue and the red, near our QTL, near that location which we've already defined. And the ones where the breaks are far away, we just discard them. We take these individual mice, and we breed them to get a population. We breed them against the purebred highs to get a population. And now we phenotype them. And the question is, well, which ones are going to be high and which ones are going to be low among these variant animals? So this is sort of what it looks like. Here is the region of our pain gene, of our pain1 gene, that we said is somewhere in the middle of chromosome 15. And these are 9 different animals that have this crossing over. Each one of them, we made many babies so that we could get a clear measure of the pain phenotype. So you can see that the first part up till here, in this particular animal, number 1087, up till here, he has the genes of, or the sequence of the F1 parent. And from here onwards, he has the genes of the high C3H parent. And so for the same with this one. Here you can see where the breaks are. And that approach allows us really to map this region here and to ask, where does let's say that these animals are all high and these animals are low, where does the break come between high and low, if that's what the phenotypes say at all? Here are the results. So these top ones here had very high pain scores, and these low ones had very low pain scores. So there was, in fact, a break where we predicted there ought to be one. And that allows us to define this interval as the place where our gene probably lies. It's probably not in this region at all, but probably in this region. So we've narrowed down the interval within which our gene is likely to be located. The same approach was done with...
Just a question. You have somewhere between 68%, a third. So
Yeah.
Is that accounted for by environmental differences, or how do you explain that?
And the relatively small number of animals, we're only talking about 10 or 15 animals, so there is variability. And also, I'm not saying that this gene counts for 100% of the effect. It has a strong effect, but it's not all the effect. Okay, so we've narrowed the range, and using this second method, RISC, we narrow the range, but in a different approach, a different method for also narrowing the range. And with the RPT, we narrowed the range to this interval here. That's what I just showed you. With the RISC, we narrowed the interval to this range. And if we just overlap them, we can say with a certain amount of confidence that our gene probably lies in this smaller interval that is common to the two methods, to the two fine mapping methods. So it's probably in there somewhere. And in there still has too many genes. There are 155 genes which lie within there, but still it's a big improvement from the 800. But we still have a way to go. How do we choose among the 155 which gene is likely to be the one or the ones that are important. And here we use 4 different screening methods, which I'll go through one at a time. The first one says, well, if injuring the nerve causes this pain behavior to happen, we would expect that if a gene is important for the behavior, then injuring the nerve will change the expression of the gene, either make it increase or make it decrease. We can't guarantee that, but it's likely. It's a reasonable assumption. Now, we actually have, as part of a different project, we have information on the entire genome of these 2 mice strains, asking about expression using F-matrix expression arrays. So this gene here, for example, galanine, which is a neuromodulator in the pain system, in this strain, in the C3H strain, it's expressed at a relatively low level normally. These are individual mice, the dorsal ganglia, sensory cells. So it's expressed normally at a fairly low level, but after a nerve injury in this strain, it's very much upregulated. There's a lot more of the protein made, or of the transcript of the mRNA made, after the nerve injury. So that would be a candidate. It's affected at least by the nerve injury. And in this other strain, in our second strain, the C58, it also is expressed at a low level, and it's increased, although not quite so much. So this would be an example of one that would meet this criterion of having its expression changed. So using that method, here are the 150. This is just a list of the 155 genes. You can see their names here. And if you can't see their names, it doesn't matter because they don't mean very much except the ones that are interesting. The ones marked in yellow are the ones that meet the criterion. So, in fact, we found of the 155, only 21 met this criterion. So we've narrowed it, although not perfect. This is one of the screening criteria. The second criterion goes like this. It says, some strains are the ones that we tested for expression, 5 strains. One of the strains is a high phenotype, that's our C3H, and 4 of the strains were low pain phenotype. So if there's a regulation, there ought to be a differential regulation. The high ones should behave differently from the low ones. So we scanned our whole 155 genes, or we queried all 155 genes, to ask which of the 155 have differential regulation, something different between the highs and the lows, between the one high strain and the 4 low strains. Again, marked in yellow, the ones that met that criterion, only 10 of the 155 met that particular criterion. And only one of the 155 met both criteria. That's this gene here, CGN, which is a calcium channel subunit. I'll say something about it in just a moment. The next screen, or the next criterion we asked about is really a bioinformatic approach. Now, we know from work of others the complete base pair sequence of the DNA of some number of inbred mouse strains. And of the ones that are published, we have the pain phenotype of 7 of them. And it turns out that 4 of them, of the ones that have been totally sequenced that we have phenotype, are low, and 3 of them are high. So we apply the following logic. We say that of the 155 genes, we want to find some that are identical, where the sequence is identical in all the lows, identical in all the highs, but different between the highs and the lows. Okay, that's, again, a logical guess that criterion may apply. So we apply that, and 35 genes meet that criterion. And finally, one more criterion we asked, and this is perhaps the weakest of all, of the 155, which ones are recognized up until now as having something to do with pain physiology? Our gene was among 35. Which ones have anything to do with pain physiology? Not all that many. Only 9 of the 155 are mentioned anywhere in the literature as having to do with pain physiology, but our gene CACGN2 is one of them. So we have 4 different criteria. Of the 155 genes, 9 genes met 2 of the 4 criteria. 0 of the genes met 3 of the criteria, and only 1 gene met all 4 criteria, and that's CACGN2. So this is our favorite, our candidate. But we want to have some functional validations, and we have several of them. For example, we immunostain for the protein, and in fact the protein is expressed in sensory cells in the dorsal ganglion, and in neurons, not glia, which is nice to know. We've also recorded electrophysiologically from injured nerves in these, in mice that have this gene knocked out, and there is an effect also on the ectopic firing of the injured nerve. I'm sorry I said that very briefly. I should explain it more, but we're running short on time. Abnormal firing in the injured nerve. So the firing normally arises from the skin or from receptors at the sensory ending. And here, after you've injured the nerve, such as an amputation, the neuroma, the stump of the cut nerve, turns out to be a source of abnormal firing. We studied that a lot over the years. That's a whole other...
That gene is...
yes, and I'll explain why in a moment. So here, in fact, you can see the regulation, and the regulation is actually larger in the high strain than in the low strain. Now, it turns out, and this is really the answer to your question, it turns out that there's a natural mutant, or at least there's a mutant that was discovered some time ago, called stargazer, mutant mouse, that where this gene is mutated. It's actually knocked out, not quite a knockout, but almost a knockout. This is a very, very important research tool in the epilepsy literature, although it was never used before in the pain literature. So we asked, well, how about our pain phenotype? And it turns out that in the wild type and heterozygotes, there's a very low pain phenotype, very low pain after you injure the nerves, while as the knockouts, there's a lot of pain behavior. So this is, again, verification that we're probably on the right track. The answer to your question was, when we study these animals, that is the knockouts, they have a lot more abnormal firing, spontaneous abnormal firing from the injured nerves than these do. So the mutation has an effect on the behavior, also an effect on the electrophysiology, which for many reasons we believe is the basis of the phantom sensation, the basis for the pain that these animals are feeling. And what we really know now is that when the stargazer, which is the protein product of this gene, is reduced, the animals tend to have more pain. It's as if this protein itself could actually be a treatment, or something that could lead us to a treatment. Okay, so the last bit was, okay, now we have a good candidate from the mice, which was selected using a genome-wide screen, agreed with some narrowing after that. Is it relevant for humans? Now, the closest pain cohort that we could find is, again, our colleague Zed Seltzer, who had collected from here, from Israel, women with mastectomy, with breast amputation because of cancer pain, where, like almost every neuropathic pain, some have pain and some don't have pain. So what he collected is DNAs from 549 women, where about half of them, or 215, reported chronic pain in a chest where the breast was removed, and 334 with no pain, where there's a particular definition of what we really mean by chronic pain. So the aim now is to do an association study to see whether polymorphisms within this particular gene, the CACNK2 gene, associates or differs, using that simple chi-square approach, differs between the women who had pain and the women that didn't have pain, where again the surgery was very similar, the injury was very similar. And the result is as follows. We chose 12 SNPs within the region of this gene, some of them in exons, that is parts of DNA which are expressed in the mRNA, and some which are outside and perhaps had a regulatory role. And of those, the 12 SNP haploids, looking at all of them together, gave us significant linkage, just barely after correction for multiple testing, but did give us significant association with the pain phenotype. When we look at the individual SNPs, 5 of the individual SNPs on their own gave a significant linkage. And if we consider the ones, these 3 right here, then this particular haploid, this combination of markers, gives us quite a high likelihood and quite a high ability to predict which woman will develop chronic neuropathic pain after the mastectomy and which won't, with an odds ratio of 1.7. Now, 1.7 isn't a huge number, so if 1% develop pain, then if they had this particular genetics, 1.7% would develop pain, not a huge difference, and we wouldn't change anything in practice. However, this pain, as you just saw, is not something that happens in 1% of women. It happens in something like 30% of women. So if you're now able to predict an additional 20% that are likely to be at risk, then that could be actually useful as a prognostic tool and could tell the surgeon, well, be more careful with the surgery damage fewer nerves or do other things which may tend to reduce the likelihood of pain developing. Just to close, who is this gene? And a lot is to be said about the gene because the gene has been studied a great deal in the context of epilepsy. It's a calcium channel subunit, GABA2 subunit, calcium channels that are expressed in neurons. But that's by homology. It turns out that in terms of its function, it turns out to be what's called a TARP, or a transmembrane AMPA receptor regulatory protein. It's responsible for trafficking AMPA receptors, which are probably the main type of glutamate receptors, where glutamate is the main excitatory neurotransmitter in the nervous system. It transports or is involved in the transport of AMPA receptors from the cytoplasm to the membrane. And if there's less of this TARP around, this stargazing around, less sodium channels, less AMPA receptors are in fact expressed on the neuronal membrane. And as I mentioned, it has been implicated in epilepsy. Now it's implicated in neuropathic pain. And in fact, we have other reasons to believe that neuropathic pain and epilepsy have fundamental physiological properties in common. So we've used a new method, or at least a newly applied method, I could say, to discover what we hope is the right gene which actually accounts for this phenotypic difference in the mice and is related at least as one pain phenotype in women. And I think that we've made progress, or potentially made progress, discovering a new, or perhaps even opening up a new chapter in pain biology. What exactly are the role of TARPs in chronic pain, which has never been asked before, to the best of my knowledge. And perhaps we have even something that can be applied clinically as prognostic now. And if not, perhaps it will lead us to potential therapeutic avenues in the future. So nothing left but to thank my various collaborators here at Hebrew University and elsewhere, and to thank you all for attention.